Hello, I'm Harry Selker, and welcome to the 2021 Top 10 Clinical Research Awards. I'm joining you from my office. Like many across this nation, the Clinical Research Forum has adjusted to maintain the health and safety of all our award winners, our guests, and the communities you serve. And I am honored to be part of this wonderful event. As chair of the Clinical Research Board of Directors, I am proud to be able to support the efforts to promote understanding and acknowledgement of clinical research and its impact on healthcare and the health of the public. There is perhaps no better time than during this terrifying pandemic to highlight the importance of clinical research. Unlike previous generational disasters for which armies, relief efforts, and policy changes were needed, for this generational catastrophe, the answer can only come from clinical research. The world is depending on this work and on the people who do this work. So we are proud to highlight these efforts with you today. Now, please welcome Clinical Research Forum Vice Chair, Dr. Herb Pardes, and the founder of the Top 10 Research Achievement Awards.
Thank you very much, Dr. Selker. I am extraordinarily proud to be here with all of you today. The Top 10 Awards honor groundbreaking clinical and translational research advances that benefit the health and welfare of people all over the world. The studies being recognized reflect the influential work by investigators at research institutions and hospitals across the United States, as well as partner institutions from everywhere. There has never been more demanding eyes on clinical research than we experienced this year. The pandemic is an unprecedented event in our lifetime. And it is because of researchers like you that we are able to find solutions to not only this complex virus, but many other diseases. From the lab to clinical trials and to the approval of 3 vaccines, the pandemic placed a much greater spotlight on trusting science and the advancements that we are able to achieve. This is why we value the Top 10 Awards. They put a spotlight on excellent work that is making a tangible impact on the health of the community. Now let's get the awards underway, and I'd like, for that purpose, to welcome our Master of Ceremonies and Top 10 Chair, Dr. Albert Reese. Al?
Thank you, Dr. Pardes. It's a pleasure to be joining you today. During this portion of our program, we'll recognize each Top 10 Award recipient and congratulate them on their achievements. We'll provide specifics on the science and some details about what went into each study, and then show a short video about each award winner. Please feel free to use the chat feature to offer congratulations to the awardees as they are honored. Our first Top 10 recipient was nominated by the University of North Carolina, Chapel Hill. It provides a therapeutic option for gastrointestinal disorders. Dr. Evan Dellon and his team led a randomized controlled trial to identify a therapy for eosinophilic gastrointestinal disorders. Their study provides a potential therapeutic option for patients for whom other treatments were inadequate. Congratulations to Dr. Dellon and your team.
Eosinophilic gastritis and eosinophilic duodenitis are rare diseases with a chronic and often debilitating course, and are due to inflammatory cells like eosinophils and mast cells that cause inflammation in the stomach or small bowel. This leads to symptoms such as abdominal pain, nausea, vomiting, loss of appetite, and diarrhea. There are no approved treatments for these conditions. We conducted the first ever randomized controlled treatment trial using AK002, also called larentilimab, a medication that works through the cyclic 8 receptor by decreasing eosinophil numbers and deactivating mast cells, helping the GI tract to heal. Subjects who received active medication had nearly a 90% decrease in eosinophil levels in the GI tract compared to those who received placebo. Symptoms and treatment response were also significantly improved with the active treatment compared to placebo. Overall, The results showed that AK002 had strong benefit for treating these diseases, was well tolerated, and should continue to be developed.
Our next top 10 recipient was nominated by Rockefeller University and provides a major addition to current resources against the COVID-19 pandemic. Dr. Christian Gaebler and his team studied blood samples of 149 volunteers who recovered from COVID-19. The study provides important information assessing antibody responses to the many vaccines under development. Congratulations to Dr. Christian Gaebler and your team.
We were nominated for this prize with a publication that was published in early June in Nature that described the convergent evolution of anti-SARS-CoV-2 antibodies in individuals who recovered from COVID-19. The way we studied this immune response, it all starts with a single blood draw. So we were able to enroll over 150 individuals here at Rockefeller University from April into early May, which was not an easy task because back then there was the peak of the pandemic in New York City and the healthcare systems were at capacity and was very difficult. And we then assembled this team of physician scientists, virologists, immunologists to all work together and to tackle this problem. And the method that we used initially is we take the blood from those recovered individuals and we're looking for antibody-producing immune cells. And the way we do this is we take a small fragment of the virus, we label it, and then we use this as bait to isolate those anti-SARS-CoV-2 specific B cells. And then we use a cell sorter, which is a quite impressive machine. It's able to isolate individual B cells into small, tiny Petri dishes. And once we have them isolated, we can look at the genetic information of these cells. And throughout the summer, we went through preclinical testing, and by the end of the year, we were able to start a phase 1 first-in-human clinical trial to test those antibodies. And since we were able to license those antibodies, so they have a good potential to be able to reach patient care and be used as a prophylactic or therapeutic medicine.
Our next top 10 recipient, nominated by Weill Cornell Medicine, developed an innovative approach for detecting blood-borne tumors. Through this study, Dr. Dan Landau and his team demonstrated that their approach allows doctors to detect tumor DNA, even when it's much too sparse to be detected by existing methods, and many months or even years before recurrence is evident in imaging studies. This work will pave the way for ultra-sensitive cancer detection, creating transformative cancer treatments in which therapeutic optimization is guided by real-time monitoring of tumor burden. Congratulations, Dr. Landau, and to your team as well.
A central pillar of modern medicine is the ability to monitor the effectiveness of our interventions. And yet in cancer, there are large swaths of time throughout the patient's clinical course when we're operating in the blind. Recently, it has been discovered that tumors shed small part of their DNA into the peripheral blood. That circulating tumor DNA offers the possibility of monitoring tumors non-invasively through a simple blood draw. The challenge is that this is a very sparse signal. We have rethought this problem, and using advanced machine learning, we scan the entire genome for the tens of thousands of mutations that are found in human cancer. This allows us to track responses in real time in patients treated for metastatic melanoma with immunotherapy, and also to identify patients treated with surgery for lung cancer or colon cancer, and identify those individuals that have potentially lethal residual disease that was left behind after surgery to stratify this individual for additional chemotherapy or immunotherapy.
Our next top 10 recipient was nominated by Washington University School of Medicine. It highlights the need for a more comprehensive understanding of mechanisms underlying malnutrition. Dr. Jeffrey Gordon and his team conducted a study of duodenal microbiome and undernutrition, and were successful in reclassifying our understanding of gut physiology in children with stunted growth. Congratulations to Dr. Gordon and to your team.
Environmental enteric dysfunction, EED, is a disease of the small intestine that is thought to be a risk factor for growth stunting, a condition that affects over 150 million children. EED leads to disruption of the intestinal barrier and loss of absorptive surface area. However, since most children in low-income settings do not have access to endoscopy, and there are a lack of validated non-invasive biomarkers, the etiology and prevalence of this condition is largely unknown. The Bangladesh EED study, BEAD, was designed to identify biomarkers of this condition and assess the relationship between the small intestinal microbiota and stunting in 12 to 18-month-old undernourished children. In this study, children whose stunting was refractory to a nutritional intervention were eligible for endoscopy with collection of duodenal biopsies and aspirates. Using culture-independent methods and aptamer-based proteomics, we found that the abundances of several duodenal bacteria from children with EED were positively correlated with biomarkers of intestinal inflammation and negatively correlated with linear growth. Finally, to assess causality, we colonized germ-free mice with a consortium of duodenal bacterial strains obtained from stunted children from the BEAD study. Compared to control mice, those that were colonized with the BEAD consortium developed an enteropathy similar to that of the children with biopsy-confirmed EED. Understanding the mechanisms by which members of the small intestinal microbiota affect mediators of linear growth will facilitate the development of new therapeutic approaches for stunting, including those that are designed to reconfigure the small intestinal microbiome towards a healthy state.
Our next top 10 recipient was nominated by Stanford University School of Medicine and NYU Grossman School of Medicine, and compares two standard approaches for ischemia treatment, invasive and conservative therapy. The work of Dr. David Maron and Dr. Judith Hutchman and their team provides patients and physicians better clarity when determining the proper treatment plan for quality of life. Congratulations, Dr. Maron and Dr. Hutchman and your team.
The Ischemia Trial was designed to study whether adding invasive procedures to medical therapy improved outcomes for stable patients with coronary artery disease and markedly abnormal stress tests. The invasive procedures were an angiogram to take pictures of the coronary arteries, inserting a stent to open narrowed arteries, or bypass surgery. We found no overall difference between the two groups in cardiac event rates, but patients with chest discomfort or angina at baseline, assigned to the invasive group, had more improvement in quality of life.
Our findings show that we do not need to rush patients with coronary artery disease to undergo procedures before a trial of good medical therapy. Our results do not support the common practice of doing these procedures in patients with mild or no symptoms. The trial highlights the importance of shared decision-making between physicians and patients and should result in less stenting and cardiac bypass surgery performed.
Our next top 10 recipient was nominated by the Massachusetts General Hospital and Boston Children's Hospital and provides major advances for children with idiopathic hypogonadotropic hypogonadism, also known as IHH. Dr. Yi Min Chan, Dr. Margaret Lippincott, Dr. Stephanie Seminara, and their team identified a way of determining if children with delayed puberty is due to constitutional delay or IHH. This requires medical treatment. The findings allow children with IHH to receive prompt diagnosis and treatment. Congratulations to the team of doctors.
For most people, puberty is something that starts naturally on its own. But pediatric endocrinologists like myself often see children who haven't started puberty by the typical age. A challenge in evaluating these children is that we have no reliable method to predict which children will eventually go through puberty on their own, which most children will, and which children have a rare pathological problem with the reproductive endocrine system. To address this problem, we've developed a new diagnostic test called the Peptin Stimulation test. In our study, we administered this test to 16 girls and boys with delayed or stalled puberty to determine who would respond to Peptin and who wouldn't. We then followed these children until age 18, following some for up to 4 years, to see who would eventually enter and progress through puberty. We found that 8 of these 16 children had clear responses to Peptin, and these 8 children eventually progressed through puberty. The other 8 children did not respond to Peptin robustly, and none of them entered puberty on their own by age 18. So, in this cohort of children, the Peptin stimulation test accurately predicted which children would progress through puberty and which would not. We're now planning a larger study to determine the accuracy of the Peptin stimulation test, and we anticipate that the test will be an invaluable clinical tool for evaluating and managing children with delayed puberty.
Our next top 10 recipient was nominated by the National Institutes of Health, created the first FDA-approved therapeutic for COVID-19. Dr. John Weigel and his team of researchers studied the effects of intravenous remdesivir during a placebo-controlled trial. The team's data showed that remdesivir was superior to placebo in shortening the time of recovery in adults who were hospitalized with COVID-19 and had evidence of lowering respiratory tract infections. Due to their work, the study is paving the way for more treatment options and greater access for patients. Congratulations to Dr. Weigel and your team.
The ongoing pandemic of coronavirus 2019, also known as COVID-19, represents an unprecedented public health challenge. Given the initial reports of high morbidity and mortality early in the pandemic, we began developing a treatment study for this infection even before there were any cases of COVID in the United States. The Adaptive COVID Treatment Trial, or ACTT, was the first large randomized placebo-controlled trial of an antiviral for the treatment of COVID-19. The drug we tested is called remdesivir, which is a medication that affects the virus's ability to replicate inside the cells. In our trial, we demonstrated that remdesivir was effective at treating COVID-19. The main outcome we measured was the time to recovery, which we defined as the amount of time until someone could be discharged from the hospital. Those that received remdesivir had a time to recovery of 10 days as compared to 15 days in those that received placebo. The largest benefit was seen in those on oxygen, but there was benefit in the other groups in the study as well. In the middle of a pandemic, we were able to start the study in just a few weeks. The study was conducted at 73 sites in 10 countries and ultimately enrolled 1,062 participants in just 59 days. The study has changed how we treat COVID-19. The data from this trial supported the emergency use authorization of remdesivir in May 2020 and the final FDA approval in October 2020. This was only possible given a motivated group of networks and fantastic collaborators that came together to address this unparalleled public health emergency.
Our next top 10 recipient was nominated by Tufts Medical Center and provides the first topical formulation to address skin depigmentation due to vitiligo. Dr. Rosmarin and his team present solid evidence for the efficacy and safety of a topical formulation of the Janus kinase inhibitor. The tested compound is already being used to treat the disorder. Congratulations to Dr. Rosmarin and your team.
Vitiligo is a disease where the immune system attacks the pigment cells or melanocytes in the body. This leads to white spots, often in exposed areas such as the face and hands. Patients experience decreased quality of life and social stigmatization. Despite affecting about 0.5 to 2% of the world's population, no medications are FDA approved to repigment vitiligo. We helped evaluate ruxolitinib cream to see if patients may be repigmented. About half the patients who received the highest dose of the medicine repigment 75% or more on the face. Significant number of patients also repigmented on the body as well. Patients saw a response as early as 8 weeks after starting treatment. As a topical, this medicine was well tolerated with a favorable safety profile. It is currently being tested in a phase 3 program to hopefully become the first medicine that will be FDA approved to treat vitiligo.
Our next top 10 recipient was nominated by the National Cancer Institute and provides the first treatment option to help children with neurofibromatosis type 1, a progressive genetic disorder that affects 1 out of 3,000 individuals. Due to Dr. Bridget Weidman and her team's pioneering work, selumetinib was approved by the FDA. Congratulations, Dr. Weidman and your team.
Neurofibromatosis type 1 predisposes to tumors called plexiform neurofibromas, which can grow along any nerve in the body. They frequently occur in very young children, grow relentlessly, and cause problems including pain, disfigurement, and vision loss.
In the absence of effective medical treatments, we conducted several clinical trials with targeted agents, which were unsuccessful until we identified that the MEK inhibitor, selumetinib, shrank the majority of plexiform neurofibromas in children and improved pain, disfigurement, and function. Selumetinib became the first FDA-approved drug for NF1, which is a great advance.
Hi, my name is Philip Moss. I've been on the selumetinib trial for almost 6 years now. The way selumetinib has impacted my life is greatly. My tumor used to affect my physical appearance on my neck very greatly, and now people don't even notice it. So I used to get questions all the time about what's on my neck or am I doing alright? And now I just fit in like a normal kid.
We are grateful to the NF1 community, our participating sites, and patients and families who participate in NF1 clinical trials.
Our final top 10 recipient, nominated by the Massachusetts General Hospital, provides a new treatment option for ALS. Dr. Sabrina Paganomi and her team tested a drug that combined 2 molecules, each targeting a different molecular mechanism underlying the disease. The results of this trial are currently being used to support a new drug application, and the study marks the first time that a new drug is shown to have a positive effect on both physical function as well as survival in ALS patients. Congratulations to Dr. Paganomi and to your team.
The CENTAR trial is a milestone in our fight against ALS. The project was a beautiful collaboration between a pharmaceutical company, Amylyx Pharmaceuticals, and the ALS academic community. The project was funded in part by the Ice Bucket Challenge. ALS is also known as Lou Gehrig's disease. It's a neurodegenerative disease, and people with ALS have progressive muscle weakness. This means that they lose the ability to do the things that we do every day that we take for granted, like walking and talking. In CENTAR, we tested a new investigational product called AMX35. AMX35 protects against nerve degeneration by using a combination of 2 medications that each target a different cell component, the mitochondria and the endoplasmic reticulum, respectively. The excitement is that people who took the study drug retained physical functions longer, like speaking, swallowing, walking, and they survived several months longer. This is the first drug we have tested that has positive effects on both function and survival in ALS. This is very hopeful news for people affected by ALS. We look forward to continuing this line of research and hope that this is just the beginning of new treatments for ALS.
Congratulations again to all our top 10 award winners. The Clinical Research Forum's website has more details about each study.
Thank you to all our top 10 winners. At this event, as always, I'm amazed and blown away by your work. For the second half of the program, we'll be honoring truly momentous achievements in clinical research. As I mentioned earlier, this past year has placed clinical research in the spotlight as the world waited for the fruits of our research. Under tremendous pressure, the pandemic has inspired change, innovation, and research that has ultimately answered that call. We at CR Forum reviewed the year's clinical research achievements, and we were particularly inspired by the incredible scientific achievements and efforts that led to the successful development of the Pfizer and the Moderna COVID-19 vaccines. We were inspired to create a new, dare I call it, every 100-year award to recognize extraordinary contribution and impact. The Extraordinary Impact in Health Award recognizes clinical and translational research that benefits all peoples and nations and exemplifies the promise of science and the scientific process to solve the huge problems of our time. I cannot think of more deserving recipients than the researchers at Pfizer and Moderna and their colleagues across the academic health centers in this country and elsewhere and at NIH. The rapid development of these vaccines show what clinical and translational research can accomplish at its best. These achievements demonstrate the research continuum from basic science to translation into clinical research and into safe, effective vaccines to protect human health. I have to just stop and tell you that a patient of mine recently, a businessman, was getting his vaccine, and as he sat there getting it, he noticed that tears were running down his face. I think that is a lot of how many of us feel about this work. So, Dr. William Gruber, Senior Vice President and Clinical Research and Development at Pfizer, and Dr. Brett Lee, Vice President for Clinical Development at Moderna, are here to talk about these efforts. First, please welcome Dr. William Gruber.
So thank you, Dr. Selker, and it's my great pleasure on behalf of Pfizer and BioNTech to accept one of the Clinical Research Forum Extraordinary Impact Awards. I'm greatly humbled and honored to be in the company of such esteemed and deserving individuals who have received the awards that you've announced today. Are you seeing a slide that says COVID-19 global crisis at the top?
We are.
Okay, very good. So I'll walk through these. Again, I think this audience does not need reminding of the significance of the epidemic. These are figures as of yesterday. It's hard to believe that it was over a year ago that the first coronavirus was identified, and that, in fact, it's been now a year anniversary just during this past month in March that a pandemic was declared. And here we are poised with the advances of vaccines to actually turn back the pandemic. What I thought I would do based on feedback that I've received from Andrea is, rather than spend time going into the details of the results that I think are well known, and for those of you that have had a chance to look at the paper, but more really spend time talking about, well, how did we get here? How did we actually pull something together in such a record period of time to get a vaccine, in our case, that went from concept to emergency use authorization in 9 months? It really began, for me at least, over 50 years ago. These two undergraduates that you see here had no clue that at some point in 50 years afterwards, that they would play a gratifying role in moving forward a vaccine. So the hairy-looking guy on your left with the glasses is me, and the guy on the right, who was my college roommate, is Barney Graham, who you will be hearing from tomorrow, who will no doubt explain to you in greater detail how they came to develop the concept of the pre-fusion construct of the spike protein that was born of other work that his group has done. And I expect he'll also talk to you about the nature of other emerging threats that we need to prepare for. One lesson that I would pass on to those of you with children or grandchildren in college is get along with your college roommate, because you never know how things will turn out. The work that Barney and his group did really was born from a breakthrough pre-fusion F structure that led to a new era of RSV vaccine development, something that both Barney and I have devoted a good portion of our professional careers to. And for those of you unfamiliar with RSV, respiratory syncytial virus is responsible for 40 to 120,000 children being hospitalized in the United States today, and countless hospitalizations and deaths throughout the world. And it turns out for over 60 years since the time that the virus was first discovered, we were essentially trying to develop a vaccine with the wrong form, the spent form, if you will, that's on the right-hand side of the slide, the post-fusion F trimer. Barney and his colleagues in the 2012-2013 timeframe managed to find a way to stabilize the structure. This was work with Jason McClellan and others in Barney's group in a pre-fusion state that actually represents the form that is most recognized by individuals experiencing natural infection. Most neutralizing antibody is actually to this form. I'm privileged with my interactions with Barney over the years that we now have actually acquired licensure rights to that construct that we further stabilized and is in vaccine development for maternal immunization to protect young children. But it was this seminal work that really led to the subsequent work that I think Barney will describe in more detail that allowed them to move forward with a prefusion spike protein represented for you here on the left-hand side of the slide. Now, it turns out that Pfizer and BioNTech had already entered into an agreement to work forward, to move forward influenza vaccine based on mRNA technology. And that was proceeding at a deliberate, typical pace for about 2 years when the pandemic emerged. And it was really through the courage of Ugur Sahin and his colleagues, as well as Albert Bourla, who was the CEO, or is the CEO at Pfizer, and Catherine Jansen, my boss, who basically decided, yes, there is an opportunity here based on some preclinical work that BioNTech was already doing to bring forth not just one, but 4 different constructs into the clinic, because we didn't really know, since there was no licensed vaccine, what might work best. And so without belaboring the point, there was work that really needed to be done to determine, would we be better off targeting the receptor binding domain of the subunit protein, or RBD, or would we be better off targeting the full spike protein and taking advantage of potentially other neutralizing epitopes as far as antibody response is concerned, and peptides that might drive a cell-mediated immune response and help contribute to establishment of memory, which in the end might prove important. And without belaboring the point, some of you have had a chance to follow this in real time, and you know that the early studies yielded to us antibody responses in 162B2 protein that were above those of convalescent sera, as well as a favorable safety profile, and it was thus the P2 mutated full spike protein that we moved forward with. Why mRNA? I think many of you are aware that, of course, it's non-infectious and doesn't contain other viral proteins, so it has the potential to be quite safe. It allows for a broad immune response and the potential for frequent boosting that we think may not be the case for those vector-derived vaccines. And importantly, and this is true, and I think you'll hear this from the representative from Moderna, Dr. Leave, that this technology allowed rapid development and quick production scaling. And there's little reason to be surprised then that the mRNA vaccines were among those that were most quickly developed to bring to fruition. Now here's, again, a bit more of a peek behind the curtain about what actually happens. And I find a lot of people in clinical research are appropriately focused on the evaluation in the vaccine field of getting an appropriate immune response, the nature of ensuring safety and ultimately efficacy. But there's a lot that has to go on that typically takes a matter of years. And the typical vaccine development is one of 7 to 10 years from concept to the time that you have a license. So how was this actually contracted to such a short period of time? that's represented for you on this slide, and there are two fundamental takeaways. The first one is that before there was any, even animal data that was reassuring, much less human data, a decision was made at the senior levels within Pfizer and BioNTech to go ahead and begin producing vaccine at manufacturing scale at risk, rather than wait, as we typically do, for late phase 2 evidence that you have an appropriate immune response and an appropriate level of safety to reassure yourselves and the public of safe and effective vaccine. The other key piece is really shown in the center. And again, I've been involved in vaccine development for over 35 years, and this was a new approach. And that was that we essentially conducted a seamless phase 1, 2, 3 study without pause, but with direct real-time interactions with regulatory colleagues throughout the world, particularly CBER at the FDA in the United States. And again, I can't say enough for the cooperation that was provided by the FDA to review data in real time that made this possible. Normally, there are pauses, as many of you know, between phase 1 and phase 2 to make internal decisions, to make decisions with regulatory authorities, and that's particularly true after phase 2. And that can amount to months, or in fact, sometimes years before one can move forward. I can tell you that as we made the decisions to move forward from phase 1, we essentially finished a submission of a package that we've been submitting in real time to the FDA, such that by the following Monday, we were in phase 2, phase 3. So again, real credit to the FDA. Mind you also, because we're often asked, well, did the speed in any way compromise what we would normally do? And the answer to that is an emphatic no, that no shortcuts were taken as far as safety was concerned. No shortcuts were taken as far as developing the assays and determining the appropriate immune response. And no shortcuts were taken in terms of the work that was done in phase 3 to assure ourselves about efficacy and safety of the vaccine. What was done was essentially eliminating the white space that normally exists and having everybody row together, whether we're talking about people at Pfizer, regulators, investigators, and certainly the participants in the trial. And so this ultimately led, of course, to the publication that's and the work that's being honored today, which I'm very grateful to be a part of. I'm going to just very briefly share some of the highlights of this that I think are well known to you. The first represented here is the safety profile of the vaccine. First of all, looking at local events with dose 1 shown on the top panel and dose 2 at the bottom. And I think of those of you that are familiar with vaccines can easily recognize that the safety profile compares very favorably to routinely administered vaccines. Very little redness and swelling, in fact, less than some. And the pain at the injection site, which is common in all vaccines, was mostly mild to moderate, well within acceptable safety profile. And then if we look at the nature of systemic events, I only show here the events, in the interest of time, occurring in the first 7 days after dose 2 in 16 to 55-year-olds and greater than 55-year-olds, because the reactions tended to be more common after the second dose than the first dose. And you can see for yourself as you walk across the systemic reactions in the vaccine group on the top panel compared to the placebo, that in younger adults, we had up to about a 16% fever rate. And in older adults, this tended to be lower. And the other discriminating feature, or one of the more particularly discriminating features, was chills, where you can see a 35% rate in the younger folks and 22.7% in the older individuals. So again, mostly mild to moderate, and again, in our experience, well tolerated. And importantly, upon review by the VRBPAC committee, the FDA advisory committee, and the FDA led to agreement that this was a satisfactory safety profile that could allow the vaccine to move forward. And of course, I think you're all familiar in one way or another with this data, either from the paper or what's been publicly presented on news channels. Again, the colors are reversed here from what's actually in the paper. But it was very clear that beginning fairly shortly, even after the first dose, protection was seen, ultimately exceeding 95%, beginning 7 days after second dose, delivered 21 days after the first. The red dots represent the cases seen in the placebo recipients, and the blue dots represent those seen in the vaccine recipients. I will honestly tell you, in my wildest dreams, I had not anticipated that we would actually have efficacy in the 95% range, and I think this is a credit to the nature of the platform by virtue of intracellular expression of antigen, and then we're likely getting a combination of antibody and cell-mediated immune response, which was also demonstrated in some of the earlier trials. This is important, particularly because of the efficacy that we began seeing as early as 11 to 12 days in a circumstance where we had very little neutralizing antibody, suggesting that something more is in fact at play. And of course, we'll have an opportunity over the longer term to see how effective the vaccine can be and what proves to be important. Again, the efficacy that was seen was gratifyingly high across all age groups, all racial and ethnicities, and also across geographies. We conducted the study in a number of different countries and did not see differences there. Also gratifying that the efficacy seemed to be well represented, whether individuals had underlying risk conditions or not, conditions like cardiopulmonary disease, renal disease, diabetes, and obesity. So that led, of course, to what you know, an emergency use authorization indication. For those of you interested in the details, you can see the link down at the bottom of this slide. But suffice to say, this vaccine is now dosed at 30 micrograms, 2 doses given 21 days apart. And I think you're well aware of the presentation and the nature of the cold chain. Gratifyingly, in the United States alone, there have been over or close to 73 million people now that have received the vaccine. And based on feedback and from the CDC and the FDA and our own pharmacovigilance, the safety profile remains favorable. And again, I'm going to just finish up here with a few slides to just point out the time course of this, because it is striking that, as you see on the left-hand side, it was when the agreement was struck. You can march your way across the site, seen first in human in May, but by July we were in phase 2, 3. By November, the efficacy and safety endpoints were met. By shortly thereafter, we were filing and gaining emergency approval in the case of U.S. emergency use authorization. And as of March the 26th, we now have over 212 million doses shipped throughout the world. And as you can see in the text at the bottom of the slide, the vaccine is authorized or approved in 68 countries as of March the 22nd. Our work is not yet done. We obviously want to continue to ramp up production to serve the global need. We're looking at new dosage formulations that can be more refrigerator stable. We've begun studies to look at boosting for assessing durability of response and the ability to cover variants. We've done studies in pregnant women and in children, and are moving forward soon in immunocompromised populations. And of course, I would be remiss if I didn't make at least some comment about preparing for the potential for emerging SARS coronavirus strains that have the potential to escape protection. So I think you are familiar with the nature of 3 particular variants of concern: the B117, originally identified and related to the UK, B1351 from South Africa, and P1 from Brazil. All of these have the potential to lead to reduced neutralizing, or rather that vaccine neutralizing responses may not effectively neutralize. And so it was incumbent upon us to look at that as we gained field knowledge as to whether or not the vaccine could protect. This data was just published within the past several weeks in the New England Journal of Medicine. On the left-hand side, you can see neutralizing antibody responses in vaccinees to the vaccine strain. And as you walk across, in general, the responses to whether we're talking about the 117 or mutations, 1351 variety or P1, are generally well neutralized, perhaps with a bit less in terms of what's seen as far as the South African strain. But I'd remind you again that it's not at all clear that neutralizing antibody itself is the coin of the realm in terms of durable protection. And I think we await seeing what actually happens in terms of field experience And for those of you who are unfamiliar, we have individuals in South Africa enrolled in the trial for whom cases are being obtained that we will be able to actually look at potential protection. Again, I want to end by just thanking all the clinical trial participants and their families, obviously the sites, investigators, and the dedicated staff, our clinical trial CRO and partners, governments and regulatory authorities, Operation Warp Speed, and colleagues at BioNTech and Pfizer, and a special shout out to members of my leadership team, as well as individuals at Pfizer that I've worked with for over 2 decades that really made it possible for delivering on some of the work that I'm describing for you today. And I just wanted to end with one final vignette, and it's often difficult for me to get through this without getting choked up, so let's see how I do. And that is that several investigators alerted me that during the course of the trial, they had individuals sporting this pin that came in to be participants, particularly the early volunteers in phase 1. And I think this is likely familiar to all of you. This was a Polio Pioneer Pin given to children who participated in 1954 in the Tom Francis supervised Salk field trial with polio vaccine. These individuals proudly wore this pin. And so it's remarkable when you think about the fact that they were volunteered by their parents. That volunteer spirit lasted with them throughout over 60 years. And here they are again, volunteering again for a potential life-saving, effective, and safe vaccine. So I wanted to particularly single out those individuals, but they stand as archetypes of the volunteers in general, in our case, over 46,000 who have given their time and effort to participate in the trial. So again, I'm very grateful to have been a part of this whole process. I'm particularly grateful to participants like the Polio Pioneers, and humbled and honored to accept this award on behalf of all of them.
Thank you very much, Dr. Gruber. That was wonderful. I now would like to welcome Dr. Brett Leave of Moderna.
Thank you so much. Well, I apologize. I have no slides, and I really am grateful for that very thorough overview and review of I see a lot of similarities, obviously, in the work that our colleagues at Pfizer did with the work we did at Moderna. Well, good morning, Dr. Selker, Parties and Lowe, Clinical Research Forum board members, and 2021 recipients of the Top 10 Clinical Research Achievement Awards. I want to thank you so much for bestowing this incredible honor upon our organization. On March 11th, we marked the grim anniversary of WHO's declaration of the global pandemic caused by SARS-CoV-2. This is the 5th pandemic since the start of the 21st century, and the 3rd in that time caused by a beta coronavirus. At this time last year, as an infectious disease specialist, working in the pharmaceutical industry to develop more effective influenza vaccines, I was compelled to contribute my skills to this new and ominous public health threat. My first thought was to join my former clinical infectious disease colleagues who are working on the front lines at Tufts Medical Center here in Boston. And I started an accelerated process of gaining hospital privileges. As vaccine candidates against SARS-CoV-2 started appearing, and with the knowledge of the limited pool of experts in the development of public health vaccines, in late April, I decided to join Moderna to help them stand up a late-stage clinical development program for mRNA-1273, as it was known at that time. The broad outline of what we at Moderna and our colleagues from Pfizer have accomplished in this past year has been well chronicled and well documented. And what I want to do today is explain what I believe enabled us to be successful and share that with you today. And I think there are three attributes that highlight this. The first is very highly effective collaborations between Moderna, the federal government, and academic research centers. Number two, Moderna's commitment to transparency. And number three, the relentless and selfless efforts of my colleagues who have worked around the clock, literally, since early 2020, and in the past beyond, and continue to do so today to expand vaccine access around the globe and to all age groups. Moderna was founded in 2010 and has established strong relationships with the US government during this time. Moderna's relationship with BARDA dates to 2016 in response to the public health emergency at that time caused by Zika virus, and then since expanded to other pandemic potential infections. The collaboration between Moderna and the National Institutes of Health began long before awareness of the pandemic threat of SARS-CoV-2. For several years preceding the 2019 pandemic, Moderna's R&D teams worked with Drs. Barney Graham, already mentioned by Dr. Gruber, Drs. John Mascola and Tony Fauci at NIAID to develop strategies to defend against pandemic threats, including the Middle Eastern Respiratory Syndrome and Nipah virus. This enabled the rapid preparedness necessary to create a vaccine candidate against SARS-CoV-2 back in January of 2020. The scientific understanding of this emerging threat was still forming, and to enable us to initiate the first in-human clinical trial with a clinical GMP batch of vaccine at NIAID in March. The collaboration with NIH continued during the spring of 2020 as Moderna began preparing for a large clinical efficacy trial. The relationship with the NIH enabled Moderna to utilize their extensive clinical trial site network to supplement our own. And it bears mentioning that the readiness and capability of these study sites, many of which are at academic medical centers, can be attributed to the advocacy of this body, the Clinical Research Forum, and the efforts of many of the people attending this meeting today. Upon initiating our first in-human trial on March 16th of 2020, the whole world was keenly aware of the clinical studies that we were conducting with this vaccine. This level of public interest and scrutiny in the conduct of ongoing clinical research was unprecedented in my memory and in recent history, prompting legitimate comparisons to the 1954 polio vaccine field trials. Despite the devastation of the first wave of the pandemic, the public was skeptical about the massive effort underway to develop vaccines against SARS-CoV-2. Skepticism was particularly strong amongst ethnic and racial groups that were most impacted by the pandemic. Clinicians and public health officials were frustrated as well by the lack of peer-reviewed data to help guide care and inform government decisions. To address these concerns, we took several actions that altered the trajectory of the public discourse. We created an infrastructure to ensure that recruitment for our phase 3 study reflected the demographics of the population most impacted by the pandemic. In addition, we closely monitored and published online the racial and ethnic demographic profiles of study participants on a weekly basis. And we paced enrollment as needed to enable recruitment of more participants from communities of color. In addition, we published our non-clinical and clinical study results in peer-reviewed journals as soon as the data were available. And lastly, in early September, we took the unprecedented step to publish an unredacted version of our phase 3 clinical trial protocol on our company website. I believe that these efforts help to reassure the public and the scientific community of our commitment to develop a vaccine that would be safe and effective for all people. On November 16th, we announced the positive results of the Cova study, our phase 3 trial. And I have received many calls, emails, and texts of thanks since that day. One of my infectious disease colleagues, who had herself contracted COVID-19 while caring for patients during the first wave, expressed how it helped mitigate a sense of dread and doom about the next impending wave. She also shared with me a message from a nurse midwife colleague who had received the vaccine as a frontline worker in January. And she wanted to please let me know that she was weeping, that there was an idea and a way out of this nightmare. I'm sure that my colleagues at Pfizer and many of you in the audience have received similar messages in that time. At Moderna, I have had the privilege to work alongside an incredible group of professionals with extensive experience in vaccine development. And together we have designed and executed a study of our vaccine's efficacy and safety on a timescale that had never been achieved, not by a long shot. And I remain awestruck by the selfless commitment of the thousands of study participants who enrolled in the trials of this vaccine. We continue to work hard alongside our study investigators and collaborators at NIH and BARDA. And my colleagues in manufacturing continue their relentless effort to deliver hundreds of millions of doses of the vaccine. Nevertheless, there remains a constant sense of urgency in our organization, motivated by the goal of expanding access of this vaccine and spirited by our success that makes all of the challenges along the way seem trivial by comparison. I remain simultaneously proud and humbled by what we have accomplished. On behalf of the over 1,300 employees of Moderna, I accept the Clinical Research Forum Extraordinary Impact Award with gratitude. Thank you very much.
Thank you both for those wonderful talks and for joining us today. And I'm sure there are many other examples that we have of people tearing up just seeing the results of this achievement. On behalf of a pandemic-weary nation, thank you for this incredible scientific achievement. We now, Andrea, have time for a few questions and answers before going to the final part of our program?
We do. We can probably take about 1 or 2 questions at this time. So if anybody has any questions, and among our panelists, you can all unmute and ask your questions, or we can type them into the chat. We'll just give everyone a brief few seconds, about 30 seconds, to see if we have any questions. Those were incredible presentations.
I guess
it's looking like people are
awestruck.
Like people are awestruck. That's correct. So why don't we go ahead and move on with our next awards?
Well, thank you again for that. And for those of you who want to just take this in again, it'll be on our website in a day or so. I now turn the screen back over to Dr. Albert Rees.
It's my pleasure to present two of our top 10 awardees with the Distinguished Clinical Research Achievement Awards. This honor is bestowed to the 2 top studies that show creativity, innovation, or a novel approach that demonstrates an immediate impact on the health and well-being of patients. Each award comes with a cash prize of $5,000. Our first Distinguished Clinical Research Award goes to Dr. Jeffrey Gordon and his team for the study you heard moments ago, entitled Integrating Global Health with the Microbiome. To understand malnutrition and the role the small intestine microbiota plays, Dr. Gordon and his team sought to diagnose environmental enteric dysfunction, used EED as the short, a disease of the small intestine which is characterized by a reduction in its absorptive area and chronic inflammation of the bowel. A dozen young Bangladeshi children were stunted and failed a standard nutritional intervention. They also underwent endoscopy so that their duodenal microbiota could be characterized and thousands of proteins expressed in cell lining their small intestine. At the same time, they checked for circulating in their blood, these cells circulate in their blood that could be measured simultaneously. When young germ-free mice were colonized with duodenal bacteria cultured from these children, they developed features of EED in their small intestines. The results provide evidence of a causal link between small intestine microbiota, EED, and stunting. This clearly is a very important study that opens door to identify new microbial therapeutic targets. Ladies and gentlemen, let's welcome Dr. Gordon to accept this award. Dr. Gordon.
Thank you, Dr. Reese. I want to thank the Clinical Research Forum for this wonderful award, which I humbly accept on behalf of my inspiring colleagues at the International Center for Diarrheal Disease Research in Bangladesh, led by Dr. Tamid Ahmed. The very talented students, postdoctoral fellows, and staff scientists in our group in the Center for Genome Sciences and Systems Biology at Washington University School of Medicine in St. Louis, and most importantly, the courageous and committed mothers and families of the malnourished children living in the Mirpur slum in Dhaka, Bangladesh, who enabled this study to be performed. During this troubling time for humanity, seeing pressing and vexing global health challenges embraced by students in ways that combine hope, humility, empathy, courage, creativity, and a commitment to innovation gives hope for the future. Current microbiome research is addressing questions originally posed by microbiologists more than a century ago, but is doing so with new and rapidly expanding sets of tools. We are witnessing its inherently interdisciplinary nature, which provides an opportunity to forge new alliances and evolve the ways we understand the underpinning of health and the pathogenesis of a number of diseases. The public is captivated. As such, there is a need for those in this field to describe their feelings clearly, in a sober fashion, to approach bench to bedside translation carefully, mindfully, and to engage in a proactive societal dialogue about the ethical, legal, social, safety, and regulatory issues raised by this research. Again, on behalf of my colleagues, thank you so much for this wonderful award and honor.
Thank you, Dr. Gordon. And once again, congratulations to you and your team. Terrific work. The second Distinguished Clinical Research Achievement Award goes to Dr. Bridget Weidman and her team for the study selumetinib in children with inoperable plexiform neurofibromas. After a series of negative prior trials to treat the development of tumors in children caused by a progressive genetic disorder, Dr. Weidman and her team made a breakthrough. Using selumetinib, a specific MEK inhibitor, the team observed tumor shrinkage in the majority of patients and overall improvements in pain, quality of life, as well as function. Selumetinib was approved by the FDA and is now the first treatment option for these tumors. Please welcome Dr. Weidman to accept this award and to hear her remarks. Dr. Weidman.
Yeah, thank you. Thank you so much to the Clinical Research Forum for this really wonderful recognition. We are very grateful and honored, and I'm taking this on behalf of the entire NF team, which first and foremost includes the families that have gone through many clinical trials sometimes to finally see success, and who've been very, very committed to this. The NIH team that worked relentlessly to, get back with queries the FDA had. There were thousands of queries. It was at complex clinical trial and definitely a marathon and not a sprint. The participating site investigators that contributed substantially, advocacy, and also funding from the Neurofibromatosis Therapeutic Acceleration Program and the Children's Tumor Foundation. A wonderful collaboration we established with AstraZeneca moving this forward, because this was a first. This was not something they or we had done before. The FDA was wonderful working with us as well. And then I do want to thank the NIH Intramural Research Program for allowing me to conduct these clinical trials, which at some point I might have said, let's stop this because it's not getting us anywhere. But we learned a lot, and having the support to take on this difficult question, I'm very grateful for. So thank you again so much. There's a lot more work we have to do, and this award definitely inspires us all to keep pushing and work the hardest we can. Thank you.
Thank you, Dr. Weideman, and congratulations to you and your team, and congratulations also for your persistence so that you could achieve the outcome that you have. Well, now, ladies and gentlemen, let's welcome back Dr. Herb Pardis to present the final award. Herb?
Thank you very much, Dr. Reese. I am delighted Clinical Research Excellence Award. This is given to the study that best shows a high degree of innovation and creativity, also advances science, and has an impact on human disease. The particular study stood out because of its broad impact on patient care and treatment decision-making. The 2021 Clinical Research Excellence Award goes to Dr. David Marin and Dr. Judith Hochman for their study called the Ischemia Trial. Dr. Marin and Dr. Hochman's thorough review of ischemia treatment has positively changed shared decision-making between physicians and patients. Such decisions affect over 9 million patients in the United States with stable angina and has the potential to improve the management of millions of patients around the world. It is my great pleasure to welcome Dr. Marin and Dr. Hochman to accept this wonderful award.
Thank you so much, Dr. Pardis. David and I are very impressed by the trials selected as the top 10 nominees, and we congratulate the other nominees. We are very deeply appreciative of the board's selection of ischemia for the Herbert Pardis Clinical Research Excellence Award. Just want to comment that the CR Forum was founded to recognize the important role that clinical research plays in shaping treatments and cures. And this award honors one of the founding members, you, Dr. Herbert Pardis, and I want to extend special thanks to you and to the current chair, Harry Selker, who for decades has been on this journey of answering tough questions that are very practical and applicable in cardiovascular medicine. I also want to acknowledge the tremendous support I get from my husband, Dr. Richard Fuchs, and my institution, the NYU Grossman School of Medicine. Now, David Marin and I have worked very closely for over a decade, leading a large and very talented team. So it's only fitting that we share this award and partner to express our thanks. And I'm going to turn it over to David now to extend additional thanks to the so many who've contributed to this trial.
Thank you, Judy. This trial that we conducted together is a real great example of team science. It takes a village to do clinical research, and ischemia would not have been possible without the hard work and dedication of our outstanding leadership team, the trial staff, hundreds of study investigators and coordinators, thousands of patients around the world who volunteered to participate, and of course, funding from NHLBI. So sincere thanks to all, and to my wife, Dr. Benita Hornstein, for her patience, love, and support. Thank you.
Well, thank you very much. Do we still have a moment for questions and answers, Andrea, or are we?
We do. We have some time for some questions and answers. For those of you who connected directly through Zoom, I'm sorry that the Zoom chat is not enabled, but for those of you on our conference platform, the chat is enabled, and we are happy to accept Q&A through that. And also from any of our esteemed guests and panelists, you can unmute yourselves and ask any questions that you would like. Thank you. Harry, to see if we have some Q&A coming in, and we did have a late question as well for Dr. Gruber and Dr. Leaf too. So the question for Dr. Gruber and Dr. Leaf is, what do you see as lessons learned for influenza from your work on the COVID-19 vaccine?
Maybe I can start, because obviously one of the main reasons we got involved with BioNTech in the first place was an interest in influenza. I think one of the challenges that we face with influenza, as opposed to SARS coronavirus, is packing all the antigen expression in that we would like to pack into an mRNA. And so I think that's one of the daunting tasks, but we went into this advisedly recognizing that as a challenge, and we think we have a technology that lends itself potentially, particularly with the self-amplifying mRNA, that allows potentially much smaller doses of individual components of mRNA to allow one to pack. As you know, currently we have a quadrivalent influenza vaccine. If one wants to think about adding other antigens as well, mRNA that will express other antigens, reducing the individual doses for each of those will be key. But we're optimistic about the potential to be able to do that in the future.
Yeah, this Brett, I would add to what Dr. Gruber said, that there are some challenges, but I think there's incredible promise. The efficacy of seasonal flu vaccines averages 50%. I think in my wildest dreams didn't expect efficacy results like we saw with this. They're both mRNA viruses, both RNA viruses. A little more infidelity on the flu side, but in terms of the reproduction and proclivity to drift. But I think we're also starting to see that this SARS virus is also displaying some properties that are quite similar to flu as well. So I think there's a lot of synergy there, and the nimbleness of the platform will enable us to respond quickly to changes in the seasonal strains as well. Thank you.
Thank you. And I can let all participants know that the chat function on Zoom has now been enabled. So if you are on Zoom and you wish to ask a question, we are now able to handle it. So please go ahead. Let's just give it another minute or so. I know from my email that there are some questions. Another few seconds, we should have some more questions in here. Thank you. We have one question. How straightforward is it to develop vaccines directed against the mutants?
Maybe I'll start, and keen to hear Dr. Gruber's remarks as well. I think this is it's a rapidly evolving situation. I think the major area of uncertainty is, in contrast to flu, where you have a well-established infrastructure for defining antigenic drift and the phenotypic distinction between viruses, and a sort of commonality of the approach. You have the WHO and FDA annually agreeing upon what strains will be in the vaccines. No such structure exists yet for SARS-CoV-2. There's a lot of, I think, effort and interest in this, and it will be necessary. But I think the biology of making a variant vaccine is actually quite straightforward. It's just deciding which variants we need to be targeting globally in this effort.
This is Bill. I'd agree with Dr. Lieb. I think, I am perhaps, I count myself among the optimistic that the very nature of the mRNA construct does so much to duplicate what natural infection does. And again, it may be a bit apples and oranges, but certainly from my experience in the flu field, when we were dealing with live attenuated viruses versus inactivated, one of the things that we saw was an increased breadth of response to drifted strains in the circumstance where you got intracellular expression. And so I can't tell you at this point, as I said, we're going to get data, I think all of us, either from what's happening in nature or from controlled trials about whether South African strain, for instance, are now appropriately labeled 1351, so as not to impugn South Africa, whether that will actually be what we often refer to, at least internally at Pfizer, as that ultimate nasty variant, the one that does have the potential to escape. But obviously, the nature of the mRNA platform, because of its speed, does lend itself, if there is a nasty variant out there, being able to adapt and administer it as either a primary series or as a booster dose. And so I think that's something we are going to pay a lot of attention to in anticipation of the potential for that nasty variant. But as I said, I'm sort of cautiously optimistic that at least the variants we've seen so far may not prove to be the nasty ones, and that we may be able to get by with the current vaccine. Now, again, I want to be careful here and say that the characteristics of mRNA vaccines may be very different than other vaccines, whether they're adenovirus vector or whether they're basically peptide-derived vaccines and the like. It's the nature of the platform that has lended itself to this very high efficacy that I think offers some cautious optimism that it may offer some breadth.
Thank you. We have one more question. And this again is for Dr. Lieb and Dr. Gruber. Do you anticipate that kids will tolerate your mRNA vaccine better or less well than adults?
So maybe I can start because we now are accumulating data, and I know Dr. Lieb and his colleagues are also looking at pediatric populations. We intend to, in fairly short order, submit data on 12 to 15-year-olds for emergency use authorization. We included a population of 2,000 children randomized 1-to-1. I can tell you that based on the blinded data, the reaction profile we're seeing looks very favorable compared to 16 to 25-year-olds. So that's encouraging. Nonetheless, we recognize that we have seen in general some increase in terms of reactions as we move from older adults to younger adults to the adolescents. And so, we are taking a very judicious approach to the study that we've started in 5 to 11-year-olds, and are going to work our way down with dose ranging in those age groups as rapidly but as safely as we can manage. So I think, that's a long-winded answer to say that there is the prospect that the reactogenicity could be higher. I will tell you, I was surprised that the reactogenicity wasn't higher than it was in the 12 to 15-year-olds. I'd sort of anticipated that might be the cut point where we would see something that was dramatically different. So we'll see. But, the intent is recognizing that obviously vaccinating children has the potential, particularly for public health significance in terms of reopening schools, allowing individuals to be comforted in the schoolroom setting, particularly for teachers and others caring for those children. So we're excited about the prospects for that.
Yeah, I don't have much to add except that we are obviously conducting similar trials. We are we've enrolled our adolescent trial fully and hope to have results later this year in regards to both the safety and the immunogenicity of the vaccine in that group. And we've also initiated a trial in children down to 6 months of age, a dose ranging age de-escalating trial that just began enrolling a little over a week ago. So we're agnostic, how it will play out, but I think the likelihood is that things will be okay and we'll be able to find the right dose for children.
Thank you, Dr. Leib and Dr. Gruber. I'd like to now turn it over to Dr. Rees, who has a few words to say, and then Dr. Harry Selker will close us out. Dr. Rees?
Thank you, Andrea. So let me just make some very brief remarks serving as chair of the Top 10 Awards program. This is a special honor for me. It's sort of like going to the symphony and enjoying the harmony of the music. And listening to first of all, reviewing the top 10 award submissions is quite a chore, but is it one of those enjoyable chores where you're going through a number of exciting applications and abstracts. But I think listening to each of you make presentation has just been even more exciting. So first of all, let me congratulate all awardees on on spectacular work this year. Each year, it's more and more exciting. We want to, as the Clinical Research Forum team, we want to affirm and celebrate great work, investigators, clinical and translational work every year. So my final appeal to you is keep an eye out for great work every year. I encourage you to nominate colleagues or nominate work that you're aware of. We want to celebrate our colleagues' work or celebrate our own, but make certain that we do that because we think as a consequence, we not only are helping individuals to continue to aspire to higher heights, but also we're helping our population, our colleagues. So once again, congratulations, spectacular year. Keep these nominations coming. Give us the joy of reviewing these abstracts every year. So with that, my colleague and CR Forum Chair, Dr. Selker, it's all yours.
Thank you, Al. First of all, thank you for the tremendous researchers who have shared their work with us today. To continue Al's analogy, it is sometimes you go to the symphony and the music just flows over you and it's overwhelming. And that was the totality of this was really remarkable. Thank you, Dr. Rees, for chairing this year's Top 10 competition. As you got a sense, it is a huge undertaking. Dozens and dozens and dozens of articles are reviewed and evaluated repeatedly. And also to Dr. Herb Pardes for his passion, dedication, and support of great research. Please join us soon at 1:15, I guess, at Translational Science 2021, and also again at 3:15 to hear all of the top 10 researchers present their studies in more detail. This concludes our award program, and thank you for carving out time in your schedule to recognize this great work in clinical research. To all of our colleagues out there, stay safe, stay well. Thank you very much.